All right, guys, so you will, as I'm going through this presentation, you should take notes on what the vocabulary terms are and try to put them into your own words. And that will be an assignment that you submit at the end. So I will go over kind of the vocab, but try to put it into context as well. Really focus on making sure that you understand what the terms mean, and don't worry so much. I'm going to give you some examples of them. You don't need to memorize the particular details of those, but without further ado, let's go ahead and take a look at some genetics vocabulary. Um, Pardonnez-moi. So the reason, actually, let's start with that. The reason that we're starting off with genetics, and it seems kind of out of place, is because you really have to understand some of these terms that relate to chromosomes and what genes are, to be able to look at how the cell cycle is regulated, which leads to a lot of cancer implications. So as you probably found in your research, a lot of signaling cascades are going to end up with either the expression of particular proteins that can lead to cancer or with cell proliferation. So, all right, let's start out by talking about the genome. No, not that one, this one. So this is an image that is taken of all of the chromosomes that we have. So a genome is all of an organism's genetic information. And in the case of what you're taking a look at here on the left, that is the human genome, which actually was only sequenced the early part of this century, like I think 2004 or 2005, just so that you guys know how old I am. I may have been still in high school at that point. Anyway, so the human genome was sequenced, and surprisingly, scientists found that of the two sets of 23 chromosomes, which they already knew that we had, there were only about 20,000 genes, which was way fewer than what was expected, and about 6.5 billion letters. So I'm going to really talk a lot about what is a chromosome, what is a gene, and what am I talking about when I say those letters. All right, so genomes, chromosomes, and genes. Here we have DNA. You have seen DNA. I hope we talked about it at the beginning of the marking period or the beginning of the semester. Hello, world. And we know that the DNA is going to be made out of nucleotides. We're going to try to keep this nice and simple and not go into the detail quite yet, but we do need to understand that there are 4 letters of these DNA sequences, either A, T, C, or G, and each nucleotide is going to be either an A, T, C, or G. And then when I talk about things like base pairs, I'm looking at both sides of the DNA. So again, 3.5 or 3.5, 6.5 billion base pairs in our genome. So that's a really big molecule. Now, those are not all in one chromosome, obviously. Those are split up among the 46, but nonetheless, that is a really, really big molecule. Each one potentially up to about 400 million monomers long. All right, so I mentioned chromosomes, and the reason that we have chromosomes in the first place is because of the length of our genomes. There's so much genetic information that needs to go into each and every one of the cells in our body. So if you're a multi-celled organism, you're going to have a lot of cells that do not need to have all the genetic information there for their particular role, but they do need it because that gets passed along. So how do you take all of those 6.5 billion base pairs and put them into a single nucleus? And the way that cells do it is by coiling that DNA, really keeping it nice and organized. Think of this almost like if you were to just take a giant string and try to like separate it all out, it doesn't work. You have to have it like rolled up or spooled up onto something. And so our chromosomes is just the individual pieces of DNA that get wrapped around these different proteins and coiled and then supercoiled until finally they make chromosomes. Now, the problem is that when there are chromosomes present in that really condensed form, they're not very accessible for getting the information out. So for that reason, most of the time in eukaryotic cells that are not actively dividing, the DNA is going to look more like this. It doesn't quite coil up all into that super condensed form, and we're going to call this chromatin. So chromatin is just DNA that is coiled around these different proteins, but still accessible so that they can be used for their genetic information. But, all right, so what is a gene then? If we take a look at that giant chromosome or that chromatin, we see that there is different kind of segments of it, and it's really all one continuous segment, but functionally, it's got its own discrete parts. Each one of these is a gene, and it makes some kind of functional product. Back in the old days, they used to say like one gene codes for one protein, but it turns out that a functional product can be more than just a protein. For our sake, I want you to think of one gene, one protein, okay? Especially if you've not already taken bio before. Let's keep it simple, but FYI, maybe keep it in the back of your mind, is that we're finding that genes can be for proteins, but they can also be for things like RNA and other regulatory elements that are going to be found in the genome to be able to control which genes are transcribed and when. And so what makes each gene different is simply the sequence of nucleotide letters. The DNA that you take from any particular part of the entire genome is still just either A's or C's or T's or G's and a whole bunch of different nucleotides strung together. So what makes each one unique is the sequence of those nucleotides. Kind of like if you were to take a look at a book, right? If you didn't know the English alphabet at all, it would all just look the same, but there's a huge difference between, you know, Winnie the Pooh and Shakespeare. So it's all about how you put those letters together, and that will determine what the functional product that you have at the end. Okay. So we know that humans have 46 chromosomes, and we know that they have all the same genes, really. But why are you different than me, and why is there only one of me? I'm sure you're grateful for that. So we need to look a little into different versions of genes. And so for this, we're going to use the example of a particular type of cell that is found in hair follicles. And that little guy right there that I have circled is called a melanocyte. And on the surface of the melanocyte is a melanocortin-1 receptor. It is a 7-transmembrane protein receptor that is G-coupled. So here's our G protein. And in a normally functioning melanocyte, this receptor is going to get some kind of external stimulus from a ligand that's going to bind. The G protein dissociates, activates adenylyl cyclase. That's going to start ramping up production of cyclic AMP. High concentrations of cyclic AMP lead to the activation of protein kinase A, which eventually has a whole bunch of other things happen before finally we have some gene expression, which is a fancy way of saying we're going to start making some proteins. So in a normal cell that has a functional melanocyte receptor, this melanocortin-1, this pathway is going to end up with gene expression. Okay, now the thing is that not everyone has a functional version of the melanocortin-1 receptor. And so when it does work, we're going to have genes that are expressed. And when this receptor does not work properly, it shuts the entire process down, okay? That means other genes are not expressed. And one of the genes that is involved or that is produced as a consequence of the ligand binding to the receptor is going to be some of the enzymes that are involved in this cascade. In this case, excuse me, I'm talking way too fast. I know that, but you know, you can play me on slow motion. Anyway, in this case, the enzymes are going to be involved in the conversion of tyrosine through a whole bunch of other enzymes to ultimately make something called eumelanin. Okay. So Miss Unger has been droning on and you're super confused. Why does it matter? Well, in people that have a normal version of this gene, of the receptor, then they're going to have expression of the eumelanin. In people that don't have the proper receptor, in people who don't have that functional piece happening, then that pathway is shut down. And so the enzymes for making eumelanin are not produced. What does that mean? It's the difference between people who have a lot of eumelanin pigment in their hair versus those that have less. That's all. Not a big difference, right? So in the case of the melanocortin-1 receptor, the difference is just hair color. And we call these differences in the same gene. So the gene that codes for somebody's melanocortin-1 receptor can either work or not work, essentially. And so we call those different versions of genes alleles. So an allele is a different version of a gene, and the presence of different alleles means there's only one of me. And luckily for you, only one of me and only one of each of you guys for the most part, unless you are an identical twin. And so some of these alleles are coding for different traits. Yay, exciting. Okay. So let's talk about what different alleles can do. This is going to be a really important piece going into the genetics unit. When we look at different alleles, you can have them have differences in functions or normal versus non-functional, which sometimes that's a bad thing. Sometimes it really doesn't matter. You can have normal versus some kind of harmful product being made. Okay, big picture. Different versions of a gene can sometimes be bad, sometimes be good, and sometimes be lethal. Let's take a look at the growth hormone receptor. You need a growth hormone receptor to function. If you do not have a functional growth hormone receptor, then that is a lethal issue. That allele leads to death. Okay, so you must have the functional allele. You must have a working copy of that gene in order to survive. Well, I don't know if you've ever watched Little People, Big World or ever heard of achondroplasia, but in this case, these folks have one non-functioning version of the gene involved in signal reception for growth hormone. But that's the good thing about the human genome and other sexually reproducing organisms, is that we are going to have two copies of all of the chromosomes, two sets of chromosomes, one of which was inherited from one parent and one from the other. And this is going to allow for a tremendous amount of redundancy. What that means is that you can inherit a non-functional copy of this particular gene, but you have a backup plan. You have another gene that will work. You have another copy of each gene. So this is going to be really important because most of us have several different genes that are lethal because they don't work. We get the copies of the genes that don't work. But because we have the other copy, because our cells are diploid, they have two sets of all of the chromosomes and therefore two versions of each allele. We're hopefully guaranteed to get one that does work, or at least the odds are way more in our favor. And so what these are called are homologous chromosomes, and they are the chromosomes that have all the same genes, but potentially different alleles. Again, one from each parent, and that's going to allow for any kind of errors to be hidden behind the fact that we do have a functional one. And looking at how alleles work together, or how one will work and one won't, whatever the case might be, that's going to be the foundation of our genetics units coming up in the next couple of weeks. Actually, next year, after the new year. Okay, shut up, Unger. Moving on. Yeah, get to the point, right? I totally feel you. So let's do this. So big picture here is we need to understand what chromosomes are. So we've got our chromosomes that have the different genes on them, that have the different versions of the genes that are called alleles. And what is going to happen during a cell's lifetime is that the chromosomes are all going to be duplicated. And that process is called replication. So the DNA gets replicated and we end up having two copies of all of our chromosomes, kind of essentially making 92 different pieces of DNA, okay? Those are then going to undergo condensation or coiling, however we want to put that together, but they're going to kind of coil up really, 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 really tightly right before a cell divides. And what we end up with here are what are called sister chromatids. Sister chromatids are different than homologous chromosomes. Sister chromatids are basically photocopies of the genetic makeup. So whatever homologous chromosomes you have, now you're going to end up with two identical versions of each one of those chromosomes. So the sister chromatids are photocopies of each other, exact same genes, exact same alleles, whereas the homologous chromosomes are going to have all the same genes, but potentially different versions of the genes, potentially different versions or alleles of each gene, okay? So we've got the genetic information that's all loosey-goosey in the chromatin, which then is going to replicate, which is then going to make sister chromatids. And we just happen to know that we have 2 copies of each chromosome because we are diploid. And that means that in addition to having sister chromatids before everything gets kind of separated out, we also have like another copy of each one. Okay, so what we're going to be looking at in this unit is cell division. And cell division is really not much more than just chromosomal sorting, right? You've maybe heard of mitosis, and it's nothing more than this. I like to give the example of a deck of cards because it's pretty close to how many chromosomes we have, right? 52 cards versus 46 chromosomes, whatever, we get the idea here. And so those cards are going to double. That's happening when your cell is just doing its normal thing. Now, what happens if you shuffle those cards? If you shuffle them and then just do a break, odds are you are not going to end up with 2 complete sets of all the chromosomes that you need. You're not going to get a full deck of cards here and a full deck of cards here. You might end up with 5 or 6 aces in one of the decks. You might end up with just 2 or 3 in the other. And so cell division is all about taking these chromosomes and sorting them out properly. For that reason, it's important that we do understand what a chromosome is, what sister chromatids are. Sister chromatids are nothing more than the deck of cards that has been perfectly replicated. It is identical to the original. We need to understand what homologous chromosomes are, which are just same genes, but potentially different alleles. So if we took a deck of cards and then took another that maybe was like Paw Patrol and had puppy prints instead of spades, you know, that's kind of the idea. Okay. I'll keep going a lot more into it. And then finally, so why do we care about cell division? What does all of that have to do with what we are talking about right now? Well, first of all, cell division is going to be a major response of cell signaling. Remember, all cell signaling is going to have a reception, transduction, and some kind of response. A lot of times that response is cell growth, okay? And that is incredibly tightly regulated. If it goes all wonky, bad things can happen. So if you have errors in that regulation, it could either mean that you don't have enough cell growth and it can suppress the cell's survival, and that's a dead cell, which sometimes is good, but sometimes not. Or on the flip side, it can upregulate the amount of cell growth that occurs. That overstimulation can lead to tumors. And those tumors, if they break apart, can then lead to cancers. So being able to understand the cell cycle as a whole and how the chromosomes are separated is going to be kind of our basis for then building onto this and understanding how the cell cycle is regulated in context of all of this information with cell signaling. So I know that that was a lot of information. So take a second and go back through and see if you have now defined the following terms. For our vocab, hopefully you have a general definition for a genome, for a chromosome, for genes, base pairs, and an understanding of what the term diploid means. In terms of chromosomes, we looked at them in a few different contexts. We looked at chromosomes that could, and I forgot one, there we go, cell division too. So we looked at chromosomes in terms of when it's all loosey-goosey, that is your chromatin. We talked about sister chromatids, which are identical, same genes, same alleles. We talked about homologous chromosomes, same genes, potentially different alleles, which you would have received from one set from each parent. And then in terms of the genes, we looked at different versions, which are alleles, and those different versions can then lead to differences in the trait that is being expressed. So I know that that was a ton of information, and if you've got any questions, I will be on checking my email throughout the period because I want to make sure that you're solid there. And don't forget to submit your assignment so that you have a list of what the vocab terms are as best as you are able to define them. And I will see you guys tomorrow.